Alright, so our next video we are going to cover how viruses replicate within a eukaryotic cell or an animal cell. And the example I'm going to use here is going to be an enveloped virus. There are eukaryotic viruses that are also naked viruses. Really the difference between the naked virus and the enveloped virus is how it's released from the cell. The enveloped virus is going to undergo something called budding that we'll talk about. The naked viruses are going to be a little bit like the bacteriophage that we talked about in the previous video, in that it's just going to rupture and open up the cell and release the particles that way. All right, so in terms of infection, viral infections for cells themselves typically don't mean good things for the cell, because what ends up happening, whether you have an enveloped or if you've got a naked virus, is that cell eventually ends up dying, okay? But the virus is really interested in essentially just taking over the cellular components of the cell and using that cell's metabolism in order to make more viral particles, all right? So in terms of our steps here, there are going to be 6 steps that we're going to talk about. The first one is going to be absorption, or I'm fine if you want to call it attachment. I think your book uses absorption. But essentially what's going to happen, we're going to have our virus here, and I'm going to make this an envelope virus. We're going to have spikes on the outside of it. We're going to have a capsid, and we're going to have DNA. All right. On the outside of that cell, there is going to be a receptor. What's going to happen, that spike is going to be what's recognized and is going to allow that viral particle now to attach. All right. So that attachment or absorption is going to be our first step that's associated with our cell. The next step that's associated with that is once it's attached to the outside of the cell, that virus wants to get inside. All right? So what we have for our second step is going to be penetration. All right? And in terms of that, usually what happens is that viral particle undergoes endocytosis. So the cell membrane kind of invaginates, it pockets inside, and it brings that viral particle into the interior of the cell. All right? You can also have that viral particle or that virus enter the cell via fusion with the cell membrane. If there aren't spikes attached, say, to it, if you've just got an envelope, the envelope can kind of fuse with the cell membrane because it's made of the same thing. The envelope on an envelope virus is made of phospholipids. Cell membrane is made of phospholipids. They can kind of come together, mesh, and it can enter that way. All right. Since I started with the spikes, that's the one I'm going to show you, but just be aware that you can have it go the other way also. So in terms of that, our second step, that viral particle is going to be brought into the cell. And it'll be inside of a little plasma membrane here. All right, so it's got to get inside the cell. All right, so that's going to be our penetration. The next thing that needs to happen now, that virus needs to escape from that little bubble of cell membrane. And what happens, that kind of opens up. Our capsid then kind of opens up as well. and it's going to release the genetic material. Maybe I shouldn't have drawn that so long, but that's okay. All right. So our third step here then is going to be uncoating. So we have to release that genetic information into the cytoplasm of the cell, all right? And then typically, depending on whether it's an RNA virus or it's a DNA virus, some different things will happen, okay? Typically, that genetic material is going to migrate to the nucleus, although it doesn't always have to. All right, when it's in the nucleus, then we are going to start our next step, which will be synthesis. All right, in terms of our synthesis here, this is step 4. We'll either get DNA replication. You may get something known as reverse transcription, and I've abbreviated that RT. Something I haven't talked about up to this point is that some viruses are going to have proteins that are going to be unique to making that particular virus, all right? The process of reverse transcription is going to be one of those unique proteins, all right? There is a protein known as reverse transcriptase. And what that reverse transcriptase does is it takes RNA and it makes RNA into a DNA molecule, okay? That's not a cellular process that normally goes on in a eukaryotic cell. So that virus has to bring that particular enzyme in with it, okay? So if this were, for example, a bacteria that underwent reverse transcription, it would have to bring that enzyme in with it, okay? Once it brings it in, it can be used within the cell, and that virus can start making copies of itself, all right? So it'll copy the genetic material, and it will also undergo transcription and translation. So it'll make RNA. The RNA then will be made into the protein. So in terms of that, what ends up happening, the transcription is going to happen inside the nucleus. Translation, you may have thought I ran out of room, but remember in eukaryotic cells, translation happens in the cytoplasm. So that RNA leaves, and then in the cytoplasm, that's going to start making the components that are associated with our virus, specifically any of the proteins. So if reverse transcriptase was made and was part of this, that would also be made by the cell, okay? In terms of that, the synthesis, making the components that are going to be needed for that virus. Once we've got the components, they have to be put together. All right, so what ends up happening, you're going to get that capsid, you've got the genetic material, if we've got some reverse transcriptase that needs to go along, we'll put that in too, all right? Because this had spikes, those spike proteins would also be made, and what's kind of neat about that, those spike proteins actually don't associate with the capsid itself. The spike proteins actually go and put themselves on the outside of that eukaryotic cell. And where that comes into play and why that needs to happen that way is our enveloped virus is going to bud off of our eukaryotic cell. So what's going to happen now, let me erase this here. We're going to have our cell membrane now pocket out, and this essentially, with budding, is going to happen by exocytosis. All right? We'll have our viral capsid in there. We've got our DNA. We've got our enzymes. All right? And what's going to happen now is I'll draw it with a blue membrane here. New viral particle is now going to take part of that plasma membrane that's associated with that eukaryotic cell. And those spikes are going to be embedded in the outside. We've got our capsid. We've got our genetic material. We've got any other enzymes that have to go along with it. All right? So in terms of that viral particle then kind of just buds off of that eukaryotic cell. That can now go infect another cell. All right? So in terms of replication within our eukaryotic cell with an enveloped virus, we've got attachment or absorption, we've got penetration, uncoating, synthesis. I didn't put this one in here. We're going to assemble that viral particle. Our last step then is going to be when that viral particle buds off of that eukaryotic cell. Okay? So that viral particle takes part of the host cell membrane with it. The cell membrane isn't made and isn't contained within the genetic material associated with the virus. Okay? It kind of steals from the eukaryotic cell. All right? So this is how replication is going to happen with an enveloped virus in a eukaryotic cell. Keep in mind, if you have a naked virus, if we draw one like this, that naked virus can still attach to a eukaryotic cell. And the difference comes with our naked virus. Really, it's step number 6. Okay? With the naked virus, instead of budding off, that eukaryotic cell is going to lyse open. And it's just going to essentially release the particles, the viral particles from the cell that way, much like the bacteriophage. All right? Hopefully that makes sense. If you have any questions, please let me know.